So after transcriptions, after this post-transcriptional modification, so this mRNA now go to the enter into the cytoplasm. So what we're going to learn for this lesson, we're going to look at the translation process that takes place in ribosome. Okay, so now for tRNA molecule, first let's look at tRNA molecule. So tRNA molecule, actually this is a shape of tRNA molecule. So this tRNA molecule have two very important regions. One highlight here, this region. This region is for the amino acid attachment site. Okay, amino acid can attach. So amino acid A, amino acid B, amino acid C, and so on. Very clear? But what kind of amino acid can attach here? Okay, what kind of amino acid can attach here? It depends on the anticodons here. Are you clear? So always remember, okay, one amino acid, okay, one amino acid can have more than one corresponding anticodon. Are you clear? One amino acid can have more than one corresponding anticodon, but one anticodon will only can have one corresponding. Okay, they only can have one corresponding amino acid. Okay, clear? Okay, so means that it depends. It totally depends on the anticodon. If the anticodon is AAG, if the anticodon is AAG, then that is a particular amino acid. Okay, so first it has the anticodon. First, they must have an anticodon. So this anticodon is a complementary binding sequence for the correct mRNA codon. I will show you guys later. So it's recognized by the amino acid tRNA synthetase. Now, even though I asked you to highlight this, but in the exam, you will forget this right enzyme. So this enzyme can recognize the anticodon and they add the correct amino acid. For example, AAG, they add amino acid A. Okay, let's say AAU, they add amino acid number 2. Can you see that? Okay, so each kind of tRNA molecule binds to a specific amino acid, and the process of binding this amino acid or link the amino acid to this attachment site require ATP. Okay. Next molecule we're going to learn here is the ribosome. So the ribosome have 4 binding sites. If you look at this here, so we have first binding site, you can highlight mRNA binding site at the small subunit. So it means that mRNA can bind to this binding site. This binding site is a small subunit binding site. Okay, and then you have 3 sites here. Can you see that? So this 3 site is termed as the E site, P site, and A site. Are you clear? 3 sites. So these 3 sites actually is for the attachment of tRNA. Okay, tRNA. So a ribosome holds the mRNA templates and monoacid tRNA. The growing polypeptide change in the correct orientation so that genetic code can be read and next peptide bonds can be formed. Okay. Okay, so next. So the tRNA molecule attach the 3 depression, 3 groove here, namely E, P, and A. So as P. P is where the growing polypeptide actually, can you see that? Growing polypeptide will be here. Okay, and A is where the amino acid tRNA delivering the next amino acid. So sometimes it just called, I will call it as a tRNA enough already for your syllabus. What's my amino acid? I will tell you guys more about this. Okay, and then E site. E site is where the tRNA that have delivered the amino acid exit the ribosome. Okay, so before we proceed, just a quick one for you guys to look at it. Okay, so the process of protein synthesis, this one I want you guys to change. It has 3 distinct stages. Translation. Okay, so the process of translations, right? The translations, has three distinct stages. So one is initiations and repeating cycle elongations and terminations process. So let us look at the first process here. Okay, now, so first, as I said, that mRNA have to bind to the small ribosomal subunit. So because we have this mRNA binding site. So once you have done already, so at the start codon here, we have the first initiator tRNA or tRNA with the methionine. Okay, right here, and then you can see that A, U, and A, C, and G complementary base pairing take place. So this one is anticodon. Okay, and this is codon. So this binding allowed, okay, the Raj subunit now come in with the E side. So P side is here, and hindrance here, P side. Oh yeah, sorry. Okay, so you can see the P side and E side. Are you clear? So what's the initiations in the translation process take place? Now I can copy down. So first, a small ribosomal subunit bind to the molecule of mRNA. So the initiator tRNA. So if you want to use the initiator tRNA, just say tRNA. Okay, with the anticodons UAC, base pair with the start codon called AUG. So this tRNA carry the amino acid methionine. So one thing very fun thing here is polypeptide. They start with methionine. Okay, but after modification, okay, so during modification, okay, in some protein, a polypeptide, okay, methionine is removed. Are you clear? Okay, so means that during the modification time, after the translation, some of the polypeptide, oh, I don't need to use methionine, so they're going to remove it. Okay, so the arrival of the ribosomal complete the initiation complex. The initial tRNA is in the P site. Now, can you see that in the P site? Okay, so the A site is available for tRNA bearing the next amino acid. So this one remain empty for the next, okay, amino acids, okay, to come in. So you can take a photo. So far, okay, not clear? Okay, so now next. Now let's look at elongations of translation. So this is part of elongation already. Okay, so it means that the proteins, okay, the polypeptide is still here, amino acid end of the polypeptide. So you can see that this is the tRNA. So this tRNA V1s to come in. Okay, so why is this project happen? So they will cleave the bond here. Cleave here and then transfer. So this enzyme involved is the write down peptide transferase. Okay, peptide transferase. So this peptide transferase will do remove the bond here, bring the entire thing join here. You can see that join. So therefore, you can see the elongation process take place. And now what happened? The ribosome read three more codon, three more letter or one more codon. So this now, this one, can you see that? This one without the amino acid, right? So move to the E side, will be released. So this one in the A side now translocated to the P side, and we wait the new amino acid. So you continue. Are you clear? Okay. So in this case, describe the process label 1. So number 1, what's number 1? So the anticodon of an incoming amino A cell tRNA base pair with a complementary amino codon in the A site. Okay, you're going to come in, goodbye. So next. So peptide transferase, write the enzyme. You must write this enzyme. You must know this enzyme. Okay, so this enzyme is called peptide transferase. So this enzyme catalyzes the formation of peptide bond between the amino group of the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. Now, this is what actually happened. Okay, wait now. Yes, what I mean here is peptide transfers catalyze the formation of peptide bond between the amino group of a new amino acid in A site and the carboxyl end of the growing polypeptide in P site. So this step removes the polypeptide from the tRNA in the P site, attach it to the amino acid of the tRNA in the A site. Okay, so generally it would be something like this.
That's the Australian one.
Okay, so we have two. So we form the bond, then we're going to cleave the bond, remove the bond. Okay, and this one will be exit the center. Okay, so the ribosome translocate the tRNA with polypeptide now in the A site to P site. So the one with the polypeptide from the A site now to E site. Okay, A site, sorry, to P site. So this also bring the next codon to the translator into the A site. Okay, so this one to E site, this one to empty A site. Are you clear? So at the same time, the empty tRNA in the P site is moved to the E site where it is released. So E stand for exit. Okay. So next, when come to terminations of the translations, it will until the stop codon. It's a good idea that you remember one of the stop codons when you want to write it. Okay. Okay. So when a ribosome reaches a stop codon, so either UAG, UAA, or UGA, so the A sites of the ribosome now accept a release factor. Okay, release factor. So this release factor, okay, is a protein. They are protein, but it's shaped like a tRNA instead of amino acid tRNA. So they don't bring in any amino acid. They don't bring in any amino acid. But what is the function of release factor? So release factor actually promotes the hydrolysis of the bond, okay, of the tRNA in the P site and the last amino acids of the polypeptide. So freeing the polypeptide and everything will dissociate and wait for the next round. Okay, clear or not, guys, so far? So I'll stop the recording. So describe the role played by the mRNA in the polypeptide synthesis. Yeah. So mRNA molecule is a copy of the gene which encodes for polypeptide. So this is general role. Okay, general role. Okay. So after it leaves the nucleus, enter into the cytoplasm and eukaryotic cell, it moves toward and attach to the small subunit of the ribosome. So what is the function? They move. Okay, very important. They have to move towards attach to a small subunit of the ribosome and then expose its codon, act as a template for translation. Okay, general role. So 2 marks here. Okay, what's the role? They encode for the polypeptide. Second, they move towards and attach to a small subunit of a ribosome. So therefore, expose its codon, which act as the templates for translations. Okay, now next. So during translations, complementary base pairing between, so this is more detailed. So the second paragraph is more detailed now. So during translation, complementary base pairing between the exposed mRNA codon and appropriate anticodons on the tRNA take place. So this is very important. They allow the complementary base pairing between the exposed mRNA codons, appropriate anticodons. Okay, so why we need? So this process actually ensure that tRNA only bring the correct amino acid to the ribosome. Okay, so this is the first, number 2, number 3, number 4. And last, we have the correct sequence of amino acid can be synthesized. Okay, with this, they can actually make sure that correct sequence of amino acid can be synthesized. Okay, so you can take a photo you need. If done, just show me thumbs up. Okay, so next. Well, how about the role of tRNA? So the role of tRNA, so describe the role played by tRNA in the polypeptide synthesis. So each tRNA carries its specific amino acid to the ribosome. So the complementary base pairing between the anticodon and tRNA and the codon mRNA ensure the sequence of amino acid product is correct. Okay, so again, so in this case, this is the general rule. Then the complementary base pairing between anticodons and tRNA and the codon ensure that the sequence of amino acid of the polypeptide is correct. So this is second. Okay. Now next. So during the elongation step in the translations based on the mRNA codon, anticodon and two appropriate tRNA molecule with their specific amino acid bind to their respective codon within the large subunit. So their role now bind to their respective codons within the large subunit of the ribosome. So one at the P site, another one at the A site. So what's so important about this? So this allowed the amino acid to be held close together to form the peptide bonds. So number 3, number 4. Okay, so what else they can do? What else they can do? What else can do? So they can actually, at those stage, they can be reused after it binds to another appropriate amino acid. Okay, so these are the role of the tRNA. So in a copy of the photo, I think you start this like this, like easier. Okay, part by part. Done? Let me know. Then I continue. Okay. Now next, this part. Okay, done. If done, then I will do a summary here, very important summary. Okay. Now, so the entire protein synthesis, what we need to know, okay, of polypeptide synthesis. So first part, you need to know what is meant by gene. Okay, so I explained already, okay, what is meant by gene. So second, you need to understand what is meant by DNA triplet. You need to know what is meant by the mRNA codon and tRNA anticodon. So what is their relationship here? Okay, now number 3. So in terms of the process mechanism, then you need to know whether it's a prokaryotic cell. If the question asks for prokaryotic, versus eukaryotic. Okay, prokaryotic versus eukaryotic. So prokaryotic generally very simple, as I said that. We start from the, sorry. So start from the, oh, sorry, I forgot the